Hello everyone. In this presentation we will learn about electrophoresis. Introduction. Electrophoresis is the movement of dispersed particles relative to a fluid under the influence of a uniform electric field. Electrophoresis of positively charged particles, cations, is called cataphoresis, while electrophoresis of negatively charged particles, anions, is called anaphoresis. The phenomenon of electrophoresis was discovered for the first time in 1807 by Russian professors Peter Ivanovich Strakov and Ferdinand Frederick Royce at Moscow State University. Theory. Electrophoresis describes the migration and separation of charged particles, ions, under the influence of an electric field. An electrophoretic system consists of two electrodes of opposite charge, anode, cathode, connected by a conducting medium called an electrolyte. The separation effect on the ionic particles results from differences in their velocity, v, which is the product of the particle's mobility, m, and the field strength, E. V equals μ. The mobility, m, of an ionic particle is determined by particle size, shape, and charge, and the temperature during the separation, and is constant under defined electrophoretic conditions. Gel electrophoresis. Electrophoretic conditions are characterized by the 1. Electrical parameters, current, voltage and power. 2. Ionic strength. 3. pH value. 4. Viscosity. 5. Pore size. Steps to gel electrophoresis. 1. Prepare the gel and place it in the chamber. 2. Fill the gel with buffer solution. 3. Remove the comb. 4. Put samples into the wells by the comb. 5. Turn on the power supply. 6. When complete, turn off power and remove gel. 7. Apply stain to gel if necessary. 8. Observation and interpretation. Types of gel electrophoresis. 1. Agarose gel electrophoresis. 2. Polyacrylamide gel electrophoresis. 3. Starch gel electrophoresis. 4. Capillary electrophoresis. Agarose gel electrophoresis. 1. Agarose gel electrophoresis is a method of gel electrophoresis used in biochemistry, molecular biology, genetics, and clinical chemistry to separate a mixed population of macromolecules such as DNA or proteins in a matrix of agarose. Agarose gels are made from the natural polysaccharide polymers extracted from seaweed. Agarose gels are easily cast and handled compared to other matrices. The gels have lower resolving power for DNA but have greater range of separation, and are therefore used for DNA fragments of usually 50 to 20,000 bp in size. The proteins may be separated by charge and/or size. Isoelectric focusing agarose electrophoresis is essentially size independent, and the DNA and RNA fragments by length. Agarose gel is easy to cast, has relatively fewer charged groups, and is particularly suitable for separating DNA of size range most often encountered in laboratories, which accounts for the popularity of its use. Agarose gel is a three-dimensional matrix formed of helical agarose molecules in supercoiled bundles that are aggregated into three-dimensional structures with channels and pores through which biomolecules can pass. Agarose gel has large pore size and good gel strength, and is suitable as an anti-convection medium for the electrophoresis of DNA and large protein molecules. The pore size of a 1% gel has been estimated from 100 nm to 200-500 nm, low concentration gels, 0.1–0.2%, however are fragile and therefore hard to handle. Agarose gel has lower resolving power than polyacrylamide gel for DNA but has a greater range of separation, and is therefore used for DNA fragments of usually 50–20,000 bp in size. The limit of resolution for standard agarose gel electrophoresis is around 750 kb. It can also be used to separate large proteins, and it is the preferred matrix for the gel electrophoresis of particles with effective radii larger than 5–10 nm. Procedure of agarose gel electrophoresis. 1. Casting of gel. The gel is prepared by dissolving the agarose powder in an appropriate buffer, ST or TBE. The agarose is dispersed in the buffer before heating it to near boiling point, but avoid boiling. The melted agarose is allowed to cool sufficiently before pouring the solution into a cast, as the cast may warp or crack if the agarose solution is too hot. A comb is placed in the cast to create wells for loading sample, and the gel should be completely set before use. 2. Loading of samples. Once the gel has set, the comb is removed, leaving wells where samples can be loaded. 3. Electrophoresis. Agarose gel electrophoresis is done horizontally in a mode whereby the slab gel is completely submerged in buffer during electrophoresis. Since DNA is not visible in natural light, the progress of the electrophoresis is monitored using colored dyes. Staining and visualization. DNA as well as RNA are visualized by staining it with ethidium bromide, which intercalates into the major grooves of the DNA and fluoresces under UV light. Buffers. There are a number of buffers used for agarose electrophoresis. Most common ones for nucleic acids include tris/acetate/EDTA, TE, and tris/borate/EDTA, TBE. The buffers used contain EDTA to inactivate many nucleases which require divalent cation for their function. Applications of agarose gel electrophoresis. 1. Estimation of the size of DNA molecules. 2. Analysis of products of a polymerase chain reaction, PCR, in molecular genetic diagnosis or genetic fingerprinting. 3. Separation of DNA fragments for extraction and purification. 4. Separation of proteins. Polyacrylamide gel electrophoresis. Polyacrylamide gel electrophoresis, PAGE, is used for separating proteins in a size range of 5 to 2000 kDa. Pore size is uniform and controlled by modulating the concentrations of acrylamide and bisacrylamide powder used in creating a gel. Acrylamide is a potent neurotoxin in its liquid and powdered forms, so not used as simply a agarose gel. Steps in polyacrylamide gel electrophoresis. 1. Preparing acrylamide gels. The gels typically consist of acrylamide, bisacrylamide, the optional denaturant, SDS or urea, and a buffer with an adjusted pH. The solution may be degassed under a vacuum to prevent the formation of air bubbles during polymerization. Alternatively, butanol may be added to the resolving gel, for proteins, after it is poured, as butanol removes bubbles and makes the surface smooth. A source of free radicals and a stabilizer, such as ammonium persulfate and TIMD, are added to initiate polymerization. The polymerization reaction creates a gel because of the added bisacrylamide, which can form cross-links between two acrylamide molecules. 2. Sample preparation. The sample to analyze is mixed with a denaturant, usually SDS for proteins or urea for nucleic acids. SDS is an anionic detergent that denatures secondary and non-disulfide linked tertiary structures, and additionally applies a negative charge to each protein in proportion to its mass. Urea breaks the hydrogen bonds between the base pairs of the nucleic acid, causing the constituent strands to anneal. 3. Electrophoresis. An electric field is applied across the gel, causing the negatively charged proteins or nucleic acids to migrate across the gel away from the negative electrode and towards the positive electrode. Depending on their size, each biomolecule moves differently through the gel matrix. Small molecules more easily fit through the pores in the gel, while larger ones slowly. The gel is run usually for a few hours, though this depends on the voltage applied across the gel. Migration occurs more quickly at higher voltages, but these results are typically less accurate than at those at lower voltages. Processing and visualization. Chemicals are used for processing of the gel and the protein samples visualized in it. A. Tracking dye. Proteins and nucleic acids are mostly colorless, their progress through the gel during electrophoresis cannot be easily followed. Anionic dyes of a known electrophoretic mobility are therefore usually included in the page sample buffer. A very common tracking dye is bromophenol blue. This dye is colored at alkali and neutral pH and is a small negatively charged molecule that moves towards the anode. Being a highly mobile molecule, it moves ahead of most proteins. As it reaches the anodic end of the electrophoresis medium, electrophoresis is stopped. It can weakly bind to some proteins and impart a blue color. Other commonly used tracking dyes are xylene cyanol and orange G.B. Loading aids. Page systems are loaded from the top into wells within the gel. To ensure that the sample sinks to the bottom of the gel, sample buffer is supplemented with additives that increase the density of the sample. These additives should be non-ionic and non-reactive towards proteins to avoid interfering with electrophoresis. Common additives are glycerol and sucrose. Starch gel electrophoresis. Partially hydrolyzed potato starch is another non-toxic medium for protein electrophoresis. The gels are slightly more opaque than acrylamide or agarose. Non-denatured proteins can be separated according to charge and size. Usually starch gel concentrations are 5% to 10% for protein separation. Capillary electrophoresis. The technique was invented by James W. Jorgensen and Kryn Dierman Lukacs, and demonstrated the capabilities of this technique. Capillary electrophoresis, CE, is electrokinetic separation methods performed in sub-millimeter diameter capillaries and in micro and nanofluidic channels. Very often, CE refers to capillary zone electrophoresis, CZE, but other electrophoretic techniques including 1. Capillary gel electrophoresis, CGE. 2. Capillary isoelectric focusing, CIEF. 3. Capillary isotachophoresis. Instruments for capillary electrophoresis. The system's main components are as follows. 1. A sample vial. 2. Source and destination vials. 3. A capillary. 4. Electrodes. 5. A high voltage power supply. 6. A detector. 7. A data output and handling device. Capillary electrophoresis procedure. The source vial, destination vial, and capillary are filled with an electrolyte such as an aqueous buffer solution. To introduce the sample, the capillary inlet is placed into a vial containing the sample. Sample is introduced into the capillary via capillary action, pressure, siphoning, or electrokinetically, and the capillary is then returned to the source vial. The migration of the analytes is initiated by an electric field that is applied between the source and destination vials and is supplied to the electrodes by the high voltage power supply. In the most common mode of CE, all ions, positive or negative, are pulled through the capillary in the same direction by electroosmotic flow. The analytes separate as they migrate due to their electrophoretic mobility, and are detected near the outlet end of the capillary. The output of the detector is sent to a data output and handling device such as an integrator or computer. The data is then displayed as an electrophoregram, which reports detector response as a function of time. Separated chemical compounds appear as peaks with different retention times in an electrophoregram. Applications of capillary electrophoresis. 1. In determination of the ions NH4+, Na+, K+, Mg2+, and Ca2+ in saliva. 2. In forensic DNA analysis and to generate a profile from highly polymorphic genetic markers which differ between individuals. 3. In ink analysis, where the analysis of inkjet printing inks is becoming more necessary due to increasingly frequent counterfeiting of documents printed by inkjet printers. 4. Intermolecular binding interactions to understand protein-ligand interactions and protein-DNA interactions. 5. Cost-effective approach to do DNA sequencing that provides high throughput and high accuracy sequencing information.